then, so we can make sure that we give enough time to present as much as we can. And I would also encourage everybody to interrupt me with questions. You're feel free to just chime in and ask questions if you need anything to be clarified. So welcome. I am Charlie Anderson. I'm an associate professor at Penn State. And I'm going to be talking along with Siobhan Braybrook, who's an assistant professor at UCLA, about the structure and mechanobiology of plant cell walls. So our goal is to give you an introduction to plant cell walls in some of their complexity, and also briefly discuss some examples of using plant cell walls to study mechanobiology. So why do I study plant cell walls in particular? I got interested in plant cell walls from the perspective of bioenergy and biofuels. We are going to run out of fossil fuels, and right now fossil energy is contributing to climate instability, as you probably already know. But solar energy is really abundant, and plants have evolved over the last 400 million years or so to be able to capture and store some of that solar energy in the form of cell wall polysaccharides. And the amount of biomass that plants generate every year is pretty staggering. It's about 185 gigatons of plant-based biomass production every year, and about 145 gigatons of that is cell walls. And right now, humans use about 2 gigatons of that biomass to carry out human activities. So that's something that we're interested in potentially expanding into the bioenergy and biofuel realm. Using the cell walls of plants rather than the edible parts of plants to generate bioenergy actually also has the potential to generate negative greenhouse gas emissions. And that's a large improvement over the current use of corn-based ethanol in the United States and other places. And that's partly because plants are able to bury carbon in the ground in their root systems, and those root systems turn over fairly slowly, on the order of centuries, and they're doing this again at gigaton scales. So they're burying carbon in the ground for free for us all the time. In addition to performing all these nice services for humans, right, plants use their cell walls to perform many different functions. So the cell wall of the plant helps determine the size and shape of its cells. It helps support upright plant growth. So this is Muir Woods out in California, where I did my PhD and postdoc. And you can see that these giant redwoods are producing, truly enormous growth based on their cell walls. The cell wall also prevents water loss from the plant. It protects it against pathogens and environmental stresses. And again, it contains much of the carbon that's fixed by photosynthesis and can serve many different purposes for human society. So this is what a plant cell looks like in cartoon form. It's a lot different from an animal cell. So I'll work from the inside out. The inside of the plant cell is largely taken up in many cell types by something called the vacuole, which is a large intracellular compartment that's bounded by a membrane. And outside the vacuole is the cytoplasm, which also contains a nucleus. That cytoplasm is bounded by a plasmamembrane. And then once we get outside the plasmamembrane, we're in a space called the apoplast, which is the part of the plant that's outside the cells proper. And that apoplast can contain two different types of cell walls. One is called the primary wall that's produced while the plant cell is growing. And another is the secondary cell wall that's produced after the plant cell stops growing. One thing that I like to remind people is that the apoplast is a metabolic compartment. It's just a little different from the metabolism that's happening inside the plasma membrane. So plant cell walls are complex, but that complexity is fun to explore. And if we think about those primary versus secondary cell walls, they contain a variety of components. So this Venn diagram is showing some of the components that are in common between primary and secondary cell walls, as well as some that are more unique to primary walls and secondary walls. So the things that are in common include cellulose, hemicelluloses, glycoproteins, and metabolites. Primary cell walls are also abundant in things called pectins, as well as enzymes and a large amount of water. Whereas secondary cell walls have less water and less pectin, but contain more of a substance called lignin, and in some plants contain silica. So how are cell wall components synthesized in the plant cell? One place that synthesizes a lot of the components of the cell wall is called the Golgi. And in plant cells, the Golgi are mobile and move around in a process called cytoplasmic streaming. And these Golgi produce components of the cell wall directly, but they also produce some of the proteins that make cell wall components. So one of the really important parts of the cell wall is cellulose, and cellulose is made by a protein complex called the cellulose synthase complex. It's shown down here in green. And this cellulose synthase complex spins cellulose directly into the apoplast, but that complex itself is delivered to the cell surface via the Golgi apparatus. Hemicelluloses and pectins are synthesized directly into Golgi and then are delivered or secreted out into the cell wall by post-Golgi trafficking. And likewise with proteins, which are synthesized back in the ER usually, they're also trafficked to the apoplast via the Golgi apparatus. Lignins are produced from components called monolignals that are transported outside the cell before they polymerize in the apoplast. And that, again, that's abundant in secondary cell walls. So cell wall composition is not uniform across plant species and tissues. One of the major divisions of plants is dicots and monocots, at least in terms of flowering plants. And dicots and monocots have different proportions of especially pectin in their primary cell walls. You can see primary cell walls and dicots have more pectin. In monocots, there's a little bit less pectin. And in secondary cell walls, again, there's less pectin, fewer proteins and enzymes, and more of this lignin substance. So there's a wide variety of different cell wall types, even between these large delineations of primary and secondary cell walls. Okay, so let's get into our parts list for the cell wall. A lot of the components of the cell wall are polysaccharides, and they're made of polymers of monosaccharides. So the monosaccharides that are common in plant cell walls include hexoses like glucose, galactose, and mannose. We have pentoses that include xylose, arabinose, and apiose. Uronic acids, which are negatively charged, they have these carboxyl groups that include galacturonic acid and glucuronic acid, and deoxysugars, amylose and fucose. So all of these together are some of the variety of monosaccharides that the plant is able to put together to form the polysaccharides in its plant cell wall. So plants are just metabolic marvels. They're able to produce a wide variety of complex polysaccharides that animals are not able to do. Okay, so let's talk a little bit about cellulose and its properties. So cellulose starts out with something really simple, glucose. And that glucose is polymerized from a nucleotide sugar, UDP glucose, into a polymer of beta-1,4 linked glucose residues. So the basic unit of cellulose is called cellobiose, and it's a repeating unit of two glucose units that are flipped 180 degrees relative to one another. It has a non-reducing end and a reducing end. And these glucose chains can be many hundreds or even thousands of glucose units long. And together, they interlink with each other via hydrogen bonding to form what we call a cellulose microfibril. And cellulose microfibrils are partially crystalline. And the amazing thing is that even though scientists have studied cellulose for many decades, we don't really know what the structure of cellulose is or what its variety of structures might be. So this is a diagram back from 1962 where they were hypothesizing that this was sort of the crystalline core of the cellulose microfibril surrounded by more amorphous or non-crystalline glucose chains, right? This is sort of like slicing the cellulose microfibril in half and looking at it side on. And then this is a paper from just this year where they were modeling different potential forms of cellulose, and cellulose 1-alpha is just one of the so-called allomorphs of cellulose, and that's common in plants. So you can see that there's not really a strong consensus on what the cross-sectional atomic structure of cellulose is, which to me is amazing and tells us that there's more science to do. The other thing I would point out about cellulose is that it's very strong in the tensile direction. It has a tensile strength that's similar to that of steel. Hemicelluloses contain a backbone of sugars, and then that backbone can be elaborated in some cases with side chains. So you can see in the case of zioglucan here, it's also a beta-1,4 linked glucan backbone, much like cellulose, but this backbone is modified with xylose, galactose, and in some cases fucose residues. Right, so these side chains prevent the glucose chains from forming the hydrogen bonding that would turn them into cellulose, and instead this is classified as xyloglucan. We also have mannans, what's called mixed linkage glucan that has glucose with different linkages than beta-1,4, and xylans, which are a polymer of xylose plus some side chains. And some of these polysaccharides are thought to interact with the surface of cellulose microfibrils and help to interlink or mediate contacts between cellulose microfibrils in the wall. The third class of polysaccharides is called pectins, and pectins contain charged residues, negatively charged residues, and can form gels in the cell wall in some cases. So the simplest form is homogalacturonan, or HG for short. Everything in the cell wall has to have a name that's at least 4 syllables long. That's the rule. And we also have 2 other types of more complex pectin, rhamnogalacturonan 1, or RG1 for short, and rhamnogalacturonan 2, or RG2 for short. So these pectins can form gels, and they are thought to form what's called the matrix of the cell wall. They're a little bit more water soluble than hemicelluloses and cellulose, and pectins are one of my favorite classes of cell wall polymers. These pectins can exist in domains that can be covalently linked to each other or to glycoproteins in the wall. And then lignin, which again is abundant in the secondary cell walls of non-growing plant cells or certain non-growing plant cells, is formed of these monolignal subunits, which are linked via free radical reactions into complex polyphenolic compounds that can help waterproof and or stiffen the wall depending on the context. So we also have proteins in the cell wall, and proteins are sort of often overlooked, although they're starting to be appreciated in more detail. Those include structural proteins, so glycoproteins that can actually interact with the pure polysaccharides in the wall via complex polysaccharide side chains. And we also have non-structural proteins. So for example, there are a set of proteins called expansins that are thought to disrupt the interactions between hemicelluloses and cellulose to allow the cell wall to expand during growth. We also have transferases that can transfer polysaccharides between different groups, hydrolases that cleave the backbones, lyases that also cleave the backbones via a different biochemical mechanism, and esterases that can remove modifications of polysaccharides. So there are a lot of enzymes acting in the cell wall, and they do this in an aqueous environment in many cases. So water is an extremely important part of the cell wall. Again, it's often overlooked and not shown in diagrams, but especially in primary cell walls of growing plant cells, water is really the milieu in which these polysaccharides are behaving and acting. There are also a wide variety of metabolites that exist in the wall, and some of these include ions. Protons can acidify the cell wall and change the pH and thus change the activity of enzymes and polysaccharides. Calcium is an important ion that can cross-link pectin. And we also have a bunch of redox compounds and acetate ferulic acid. Many other metabolites exist in the apoplast. One thing I would point out, though, is that because the apoplast is separated via the plasma membrane from the cytoplasm, where a lot of primary metabolism occurs in the plant cell, these metabolites have to be transported from the cytoplasm, generated or obtained from the environment in order to act on the different components of the cell wall. So one of the things that, one of the ways I like to think about the cell wall is that it's sort of like a desert, right? Where there are resources, but those resources are somewhat limited and have to be derived from specific sources. Okay, so this is one brief architectural diagram of the primary cell wall that was generated a little while ago, about 15 years ago or so. And in this sort of architectural diagram, we can see the arrangement of some of these polysaccharides. So as I mentioned earlier, cellulose, right, this really strong, structurally important part of the cell wall is generated by the protein complex, the cellulose synthase complex. And that complex is embedded in the plasma membrane and spins these glucan chains out into the apoplast, where they coalesce into these cellulose microfibrils. And these cellulose microfibrils are long and linear. They're actually more curved than is shown in this diagram. They tend to sort of weave throughout the layers of the cell wall. But they are fairly straight, and they're long and skinny. They have a very narrow width. They're usually about 3 nanometers in diameter, but they can be several microns in length. And they form this really important scaffolding that helps hold the cell wall together and give strength to the cell wall, but still allow for enough flexibility for growth. The cellulose in the different layers of the cell wall can be arranged in different orientations, which is sort of shown in this 3D diagram. So that cellulose is often surrounded by and interlinked by hemicelluloses, such as xyloglucan, which is shown in purple here. There's been some debate in the literature over whether xyloglucan is actually forming these long tethers between cellulose microfibrils. What we think is more likely is the case now is that the hemicellulose is sort of acting at the interface between cellulose microfibrils and helping to sort of glue them together, right? So these are the so-called biomechanical hotspots that we think exist in the cell wall and limit cell wall expansion. And then that cellulose hemicellulose network is again embedded in a matrix that contains pectins and glycoproteins. So this looks pretty complicated. This is actually about 30-fold less dense than what we think the cell wall really looks like in terms of the density of polysaccharides and other components. But you can see that there are a wide variety of potential interactions here that are helping to control how the plant cell behaves and how the plant tissue as a whole is mechanically constructed. So I'm going to stop there and turn it over to Siobhan, who's going to introduce some of the aspects of the plant cell wall in action as the plant cells are doing their thing. And maybe as we switch over, if anybody has a brief question, you're welcome to jump in.
Oh, I have a question. Hello. Oh, can you hear me?
Yes, we can hear you. Go ahead.
Oh, okay, so
sorry, Morgan.
Can you introduce yourself?
Yeah, thank you, Patience. Yeah.
oh, okay. So I can hear you guys. Okay. So I have a question. Like, what do you think? Like the cellulose, hemicellulose, or pectin matrix can help enhance the mechanical properties of cellulose microfibrils?
So the question is, what happens, or how do you think the cellulose hemicellulose interactions might aid in the mechanical properties of the cellulose microfibrils? That's a great question. One of the amazing things that was discovered a little over 10 years ago is that you can actually remove xyloglucan from Arabidopsis plants, right? So we often do our experiments in Arabidopsis, which is sort of like the fruit fly of plants. And you can take away xyloglucan entirely, and the plants are okay. They grow and they reproduce, but they actually do have different mechanical properties in their cell wall. And the thinking there is that because there's a lack of xyloglucan interacting at the interface between cellulose microfibrils, that changes those interactions in such a way that prevents the cell wall from being able to be acted upon by those expansin proteins that I mentioned, but can actually make the cell wall a little bit more compliant, right? So it's more stretchable but less expandable, which sounds weird. And there are sort of some of these non-intuitive properties, mechanical properties of plant cell walls that we wrestle with as plant biologists. But the thinking is that interaction is aiding in the expansibility as mediated by the proteins that the plant produces to control its own growth, rather than it being a purely mechanical pressure-driven process.
Okay. So it's more like shear modulus, or what kind of mechanical property it can enhance? So.
Yeah, I'm gonna punt that question until the end because I talk about shear modulus in my
cool.
example. So stay tuned. Thanks.
Okay, thanks then.
Any other questions so far from the biochemistry section? You can ask more later too. You can ask both of us questions at the end too.
And guys, as a just quick reminder, could you just let the speakers know who's asking the question so that they get to know you as well? Thanks.
Okay, so then I'll go ahead. I'll just note that I put in the chat earlier because Charlie and I are doing this together. He's going to monitor the chat box while I'm talking. So if you're more comfortable putting a question in there, he can jump in for you, or we can address it at the end as well. Okay, so now we're going to go from talking about the structure of the cell wall, which Charlie set up for you, and think about how that actually works in terms of mechanics and the influence that that has on cell growth, cell expansion, but then that really means the entire plant's growth and expansion. So let me get in here and turn on my pointer if I'm going to need it. All right. So here's another cartoon of a plant cell. We do this all the time. And in this case, I have actually the same parts that Charlie showed you in the beginning, but we're going to talk about them a little bit less in terms of the cell biology and where things are made, and more in terms of their impacts on cell expansion, which is one of the major ways that plants actually grow their bodies. So there is cell division or proliferation, but for right now, we're going to really focus on cell expansion. So you've got that cell wall, which is kind of a stiff shell. And the last picture that Charlie showed you of the complexity of the cell wall, which is really just a prediction of what it might look like. We still don't know much about all of the details of that structure. And knowing the details of the structure and all the linkages therein is actually really important for us to be able to determine or predict what we think the mechanical properties of that structure might be. So there's a lot of interesting sort of predictive modeling that's going on too, to try and say if these are the linkages and this is the density, what would the material properties be? But what we're going to do right now, which is something that's done in the field, is we're going to assume that this cell wall is really just a fiber reinforced hydrogel. And so the hydrogel part of it is really going to come, and this is a primary wall, this is going to come from the pectins. As Charlie mentioned, they're, you know, hydrophilic, so they can really sort of, you can get a lot of water in there, and they're creating this sort of body matrix gel within which the cellulose fibers and hemicelluloses exist as sort of our strong fibers. The other thing we have, though, is that central vacuole, which here is light gray in the center. And as Charlie mentioned, it takes up most of the volume of a plant cell, most plant cells. It's also really important for cell expansion, because what's going to happen, we have this sort of constraining cell wall around the outside. And into this vacuole, through osmosis, we're going to get movement of water. And as water moves into that central vacuole, it's going to swell. But at some point, the cell wall constrains it. So you're trying to pack more molecules of water into a cell, but you can't actually change your volume. And what that does, if you sort of remember basic physics, gas physics, fluid physics, it doesn't matter. If you're trying to put more volume in something and you can't, or sorry, more molecules and you can't increase the volume, pressure is actually going to build up. And so what happens is we have actually a high amount of turgor pressure, we call it, but it's really hydrostatic pressure inside every plant cell. And that's really important for sort of growth and structure. If you don't water your plants, you might see them start wilting. That's because the turgor pressure that's normally in all the cells actually gives them some structural integrity. But it also provides the force that's going to generate strain and or growth within the cell wall, allowing for cell expansion. And so you had from Anders, you had sort of an idea of sort of forces in biology and force and strain relationships. So we're just looking at that same thing here, but in an interestingly different system than you would see in animal cells. And so what's going to have to happen if we have a lot of pressure inside, we've got this turgor, sort of this cell wall that's, you know, stiff on the outside. If we want that cell to expand, we're going to have some sort of loosening or change in that material, right? Otherwise, you're just going to have turgor and the cell will sit there. So one of the things we're going to talk about are what are the material properties of cell walls, primary cell walls, and how do those change in order to allow for cell expansion to occur? So there's a couple unique sort of biophysical constraints for plant growth that I want to talk about. We're going to really focus on single cell expansion or single cell behaviors, both Charlie and I actually in our examples. But there are really interesting questions that come up when you talk about tissues as well, which we both also look at, but we might not talk about today. And that's because there's not really a lot of cell motility in plants. If you think about it, every cell is sort of bounded by a cell wall. And when you get division, you get some expansion, and then we're going to lay down actually a new cell wall between two daughters. And that means that they're stuck to each other by that cell wall. And so every cell that's being produced doesn't actually get to sort of crawl and move away in these really cool motions you can see in metazoan cells. Instead, each cell is really intimately stuck to its neighbors around it. And so what that means is if, you know, one cell is going to do something like expand, there has to be some accommodation by the cells around it as well. And so you can't just sort of do something, I guess, cell autonomously without having your neighbors physically feeling that. And we'll talk about that a little bit a little bit later. So what this sort of gives us are two questions that we'll look at right now. Well, how do you expand when you have a cell wall around you? And we're going to look at wall mechanics. And then at the end, I'm going to give you just some tastes of how the change in sort of cell expansion or cell shape in one cell might feed physically into other cells around it. So how might force be sensed? If you think one cell's growing, it's going to exert some forces on the cells around it. Well, what does that do, and how might that be sensed by neighboring cells? Any questions so far? If there's terms I use that I don't define and you're not clear with, if you can put them in the chat, that'd be great, because I'm not sure always exactly what instructors who came before have introduced. I can make assumptions, but they might be wrong. Okay, so how do you sort of grow when you have a cell wall around you? Let's talk about those cell wall mechanical changes that might happen. And in order to do that, I'm going to introduce some few basic mechanical behaviors from material science. And I know some of you may have that type of background, but all of you will not. And so in the very basic sense, we can think of materials that are sort of purely elastic, just like springs. So you could think of this as being a spring made out of metal. And this here, this gray block, is sort of a weight that I'm going to place on the end of that spring. And I think for yourself, you could imagine what would happen if I had a spring and I stick a weight on it. That spring is going to stretch, right? If the weight's strong enough. And so we can think of that stretching or strain in terms of deformation. And so if we're looking in time at the length of this spring or the deformation, you'd imagine when I put the weight on, I'll get some stretching. And when something's elastic, purely elastic, that happens pretty much instantaneously. So I add the weight, it stretches. It doesn't take time to stretch. It pretty much just goes boing, down. And as long as the force here, which is the weight, is placed on that spring, it'll just maintain that same level of stretching. And when I take it off, it'll instantaneously go back down. So this is a really simple demonstration of an elastic material. And as you'll see in a second, we can see elastic deformations in plant cells. And part of that is probably due to changes in turgor pressure. And Charlie's going to give you an example of that, I think, at the end, where he's looking at these special cells in plants called stomata that swell and shrink elastically in order to yield their sort of functional behavior. But if that's sort of the most basic mechanical property that a cell wall might give you, this elastic stretching, there can also be viscous changes. And so in sort of mechanics, we tend to put viscous elements as a little dash pot, it's called, but I kind of think of it as like a syringe with a plunger in it that's filled with fluid. And so fluids tend to behave viscously, which is why it's sort of drawn this way. And what you could imagine is if I put a weight on this, that plunger is going to pull through the liquid that's in this little dash pot. And that might not happen instantaneously because, anytime you move through water, it slows you down a little bit. There's force that's being put back on you by the viscosity of the material. And so when we see that generation, sort of the imposition of force or the weight here, we can see we don't have an instantaneous jump up. Instead, we sort of have this slow sort of deformation that happens. And that's really characteristic of viscous materials. But at some point, it deforms as much as it can and it just stays static. In this case, though, if I let that plunger pull down, the viscosity relax sort of in my system, and I take the weight off, it actually can't be recovered because there's nothing that's going to put a negative force on to pull that sort of plunger back into my syringe. Does that make sense? So these tend to be irreversible type material deformations in a purely viscous system. But in reality, we can couple those two things together. We can get viscoelastic materials. And in a viscoelastic material, if I put a weight on it, what we'll see is that in the beginning, we're going to get domination we could get some initial elastic deformation, but mostly we're just going to see this time-dependent relaxation because of the dash pot, because of the viscous element. But as soon as I let go of that weight again, now the spring can actually pull back the sort of viscous element. And so you'll still get the time dependence, but the force, the retraction force that's happening is because there's an elastic element coupled to that viscous one. We can build these up even more complicated as well. And this is actually one of the things that gets done. So we're going to look at how we make mechanical measurements of plant cells. And when we get mechanical properties or ideas of how they behave, we have to back predict what type of material model might have fit that type of behavior. And that'll tell us something maybe about what the structure of the cell wall is, how that structure might be changing to change different parts of this type of behavior. So I know not everyone has a materials background, but hopefully what you can take away from this is that purely elastic behaviors should be rather instantaneous and they should be reversible, whereas viscoelastic behaviors should require some time to happen, but they can be also reversible. That makes sense. What's interesting here is when we think about growth, so if we're talking about changing the actual volume of a cell, we need that cell wall not just to stretch because of turgor pressure, we need it to actually fix that stretchness. So we don't want it to be viscoelastic completely. We don't want it to go back once the turgor pressure changes, right? We want to actually be able to change the resting length of these elements. So that's where we start thinking about how these parameters of a cell wall might relate to growth, which is plastic, and we'll come to that in one more second. People have been looking at plant mechanics for a really long time. So I'm showing you some examples from 1957, which was before you were born, but also before I think pretty much everybody in this room was born right now. And so there's some tissue level experiments that have been done for a really long time and are still done today because they're still incredibly informative, although we have some slightly more sophisticated machines. But here what we have is just a little piece of a young plant seedling. And what they've done is they've sort of cut it, and then they're going to apply a weight to it and ask, when I apply a weight to it, how much bending happens? And bending here would be strain, and the force that's being applied would be the weight. And so you would generate stress in that tissue and you would get some strain as a result of that. So we can look in this old literature and see, well, just if you take a piece of plant organ, plant tissue, what kind of material behavior does it have? Is it purely elastic when I add a weight to it? Or is there something viscous or viscoelastic about it? So this is the data from one of their figures, where what they did is sort of they take this piece of tissue, they're going to add a particular weight at 5 minutes, which is this time here, and they're just going to look at how the angle sort of changes over time. So this is where it was sort of here at this position, and they add the weight, and what you can see, I hope, is that you're not seeing an instantaneous movement of the plant organ, right? So there is some time dependence here, which would imply some viscosity in the system, right? If it was purely elastic, it would just go thunk, but it actually takes some time. And so they go like this and they watch it over time. It's sort of, okay, here, 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 here. It's still going. It still hasn't reached its plateau, but I guess they get kind of bored at some point, who knows? And they take the weight off here at 25 minutes. And what you can see is you get recovery. And some of that happens pretty quickly, but there's some time dependence there again. This is sort of telling us maybe this is viscous, maybe it's viscoelastic because they can recover some of it, right? But what's interesting is if it was purely viscoelastic, we would think it would go all the way back up to where it was. So if we release the weight, it might slowly take some time, but it should eventually get back where it was. And they didn't see that in these original experiments. And so they actually thought that there was, when they did this, when they applied this load, there was some plasticity or potentially growth that was happening in the system. Interestingly, what sort of happened since then, there's sort of more informed experiments, but it's not because they didn't know what they were doing. It's just that we sort of have better concepts and ways of doing experiments now built on the knowledge that they had in the beginning, that they built for us in the beginning. And what most experiments tend to show now is that this non-recovery due to this experiment is probably an artifact. You could think about it, if I sort of put a weight on here, I might damage that material. And so it might not be able to come all the way back if I actually exhibited too much damage when that happened. So you can get breaking in the material that isn't based on anything except your actual experiment that you're doing that can affect your mechanical measurements. And so in reality, when people do these experiments a bit differently now, they can see that you get full recovery, meaning that these tissues really do behave like viscoelastic solids. But this is a whole organ, right? And we're really interested in cell level measurements of things as well. We still do these types of measurements, they're really important, but we also want to ask on cell levels what's going on. And so in order to think about this on a cell level, what you can imagine is that the cell wall could be represented by a bunch of different viscoelastic elements. And some water is going to come in, right? And if there's enough force that the water is applying on these elements, we could end up getting some deformation in the elements, which would cause us to, for example, swell. It might take a bit of time because they are viscoelastic, but we would swell. But the question is, of course, if I take away that pressure now, if I pull some water out in a purely viscoelastic system, we should go back down to the size that we were before, which isn't what we really want for growth to happen. So I think something I want you guys to think about is if you were going to take this system and you didn't want it to shrink back when you removed the pressure, how could you actually get it to stay at the new sort of length that the system has? And for some people, that might be a bit simpler because you think about, you have a background sort of in material sciences or mechanics. But I think in anybody's mind, you can think about it. Imagine you just have that network of polysaccharides. It stretches with turgor pressure. But what we want to do somehow is actually make it so that it stays in a particular way. So if I remove the pressure, that sort of network of polysaccharides that's stretched is not going to shrink back. So I'm going to leave that for you, and if we have time, we'll come back to that question. So we have this idea that cell walls are viscoelastic, but that really came from a lot of tissue measurements. And I'm just checking the time for us. So what we wanted to do, though, is develop ways that we could measure this actually at cellular levels, right? Not just as a bulk property of a tissue, but really look at individual cells and what they were doing in individual cell walls within a cell too. So I'm going to tell you about two types of experiments that can be done to test mechanical properties in plant cells. There are also a couple other ones that if we have some time, I can touch on for you, but you can also, ask questions of any of the other instructors or send me an email if you want to know more about some of these newer types of methods. But one method is actually to take advantage of the water movement in and out of cells and actually manipulate that by manipulating the external osmoticum so that you either pull water out and look at how cells shrink, or you put water in and look at how they swell. And that's a way that you can measure actually cell elasticity, because you're basically playing with how much pressure is being put onto the cells by the water movement inside. And so the other one that is quite common now, I would say, which my group uses, Charlie's group uses this also, we do also use the swell shrink method, is to actually use nanoindentation to do tiny, tiny material tests on living plant tissues. And so we're going to look at this as well as an example. The system we're going to use is something called a shoot apical meristem, but this is really just at the very tip of the plant where there's a stem cell population that's producing new daughter cells towards the side, and then you're going to have new organs made around the side of this dome. So we'll look at a picture of it in a second. But essentially, you've got some stem cells in the center, you've got new organs that are going to be specified to sort of grow out, which means that's where cell expansion is going to happen. So this is a picture, if we're looking at down from the top, of a shoot apical meristem from tomato. And in this case, the stem cell niche here is sort of in the center. And so the cells sort of underlying the epidermal cells that we're looking at right now would be the ones that are proliferating. They don't have too much differentiation. They're just going to produce new daughter cells. The daughter cells get pushed to the periphery by the division of new cells from the stem cell niche. And then here, actually, we have a spot where a new organ bud is forming. And it's kind of hard for you to see when we're looking top down, but you can imagine this being a dome that's sort of shaped like this. And at some spot, we've got a little bump that's coming out of that surface. And that's actually going to end up forming a brand new leaf. And so what was done in this study is they said, okay, well, with confocal microscopy, we can look at stained cell walls in living tissues. And we can use sort of the shape of the cell and the volume of the cell when we mess with the osmoticum and say, well, how much does that cell area change if I pull water out, so if I shrink, or if I force water in, if I swell the cells? And so using confocal microscopy to look at this, what they were able to do is generate these heat maps where what we're looking at is how much does the cell area change when I do a given treatment. So in B here, we're looking at when they put them in water. So water's going to move in that case, the cells are going to swell. And so the higher the number here, the more the cells swelled. And so I hope you can see that sort of around the edges of this stem cell area, we're getting more swelling. But in the center, where the stem cells would be, we're getting less swelling. And so if you thought about that, you sort of think, well, that means that the cells at the side are actually stretchier than the cells in the center, which is one interpretation of this data. The converse is to ask, well, how much do cells shrink? When I put them into a hyperosmoticum, here they used a really high salt concentration. I'm pulling water out, where do the cells shrink the most? And so the cells that shrank the most were actually above that stem cell niche versus on the periphery. And so what they ended up sort of concluding from this is that the different cells within the different areas, whether you're sort of at the sides or on top of that stem cell niche, actually exhibited different mechanical behaviors with respect to their elasticity. But remember that these cells are all connected to each other, right? So one cell's ability to swell is also somehow influenced by the cells around it. So this is sort of why on the last slide I talked about this being a measure of tissue elasticity also, not just cell elasticity, because when one cell has to do something, the other cells sort of influence that property as well. But this is one example of how you can look at elastic swelling and shrinking of cells and learn something perhaps about their elasticity. What about indentation? So I'm just checking we're still okay. So in indentation, we're going to use an AFM. And because I don't know if it's been introduced to you, actually, can somebody tell me if you've been introduced to AFM before as a method of indenting and learning material properties as a class?
Not this week.
Not in the boot camp, no.
Okay, good. So I'll do it really quickly for you. So what you can imagine is you have a very flexible cantilever, and you can change the flexibility of this. And we're going to have a tip at the end of it, which, I don't know, I'm going to pretend is this tube that's sitting on my desk, okay? And so what I want to do is I have a surface that is going to, imaginary surface here. And essentially I can go ahead and using some piezoelectric motors, I can move the AFM tip down towards the surface. And it's going to contact the surface. And I know sort of when that happens through the machine. If this cantilever is really soft, it'll bend as I'm touching my surface. And classical AFM has been used actually to image really, really small, fine structures, almost like a record needle that sort of moves along the surface of a record. It can teach you about the topography of any sample that you put underneath it, including like imaging DNA molecules and proteins. But if you make that cantilever stiff enough, it can actually provide enough force to indent the material that it's touching. And that's what we mean by sort of nano indentation using an AFM. What we're going to do, I'll sort of show you, what we're going to do is we're essentially going to take, if this is a shoot tip, this is the meristem up at the top. We're going to go ahead and take our AFM cantilever and push along the surface of that meristem and ask at different points where I indent it, how does it respond to that indentation? It's not very different from taking a spring and putting a weight on it and asking how that stretches, except we're going to indent and ask with a given force, how much indentation do I get? And from that, we can get an idea of material properties. Does that make sense? I'm seeing a few nods, which is good. Okay. So this is a sort of topography that was actually generated by the AFM. So here, this is a different plant. This is Arabidopsis, our weed, our fruit fly. But this is sort of where the stem cell niche is. And here we can see these two bumps, which are new growing flowers, actually. And so this is where cell expansion is going to be occurring. And this one is a little bit younger. This one's a little bit older. So that's sort of what those numbers refer to. And over here, what we're looking at is actually this map of the elasticities in somewhat of the meristem and of the cell walls. And so the higher this number is, the harder it was to indent the material with the AFM. And the lower it is, the easier it was to indent. So the lower this number, the more elastic the material should be, the higher, the less elastic. And so the thing I just want to point out to you here, that what we're able to see is, first, these lines here are actually individual cell walls that we can see with this technique. But also we can see that over here in this oldest primordia, which is really probably expanding the most, we can see that those cell walls are probably a lot more elastic, which is what we might expect if there's some sort of cell expansion happening. So we can use AFM in this way to get real sort of quantitative measures of cell wall elasticity within a tissue, but also actually at cell levels. And I think Charlie and I will both show you some examples of that at the end if we have time. So I think what I'm going to do, I'm going to go through this. There's just a couple slides here, and I'm going to do that so we can show you a few examples of how this helps us understand plant growth. But this question of how cells might sense who's near them is actually really important when we think about, you know, that coordination of growth. And a lot of that actually tends to happen through the cytoskeleton, which Ram would have talked to you about yesterday, or at least what we know of right now tends to happen through the cytoskeleton. So here's another old paper from '71. And this is just a section of some plant tissue. So you're looking at cell walls here. And what they did is they just took this piece of tissue and they squeezed it. So they applied some force. And what they saw is that if they looked at the histology afterwards, there seemed like there was a lot of sort of alignment of cells against the direction of compression. So if I pushed from these sides, I started having cell files that organized this way. And remember, it's not because the cells moved. It would have been that they would have divided in a way that sort of responded to the mechanical stress around them. And that's sort of what made people think, maybe the cytoskeleton's involved because it's involved in cell division. And so in a more modern experiment, people looked at that shoot apical meristem again. And what they did is they said, okay, well, let's look at microtubules, which you learned about from Ram. And let's see what microtubules look like with respect to the shape of a meristem. And so there's some modeling here that I'll try and explain briefly, but down here in green, what we're seeing are the microtubules labeled with a fluorescent marker. And when they did this, they sort of saw that between the stem cell niche here and a new organ over here, they saw this pretty strong alignment of microtubules in this sort of crease or valley. Whereas up at the top, the microtubules didn't have that strong orientation. And that's sort of pictured here in blue. And what they did is some sort of mechanical modeling to ask whether or not the shape of the meristem would generate different stresses that might align those microtubules. And the current hypothesis is that, yeah, microtubules actually do align according to stress that's applied to the tissue, either externally or by its own shape. And that somehow can actually change the mechanics of the wall. And the way that that happens, remember the cellulose synthase that Charlie told you about that makes cellulose fibers, and the fibers in the wall are actually important for mechanics. Well, the microtubules tend to guide where that cellulose synthase complex goes. So in the cases where I have a crease here, if my microtubules are all like this, my new cellulose fibers will also be very oriented in a particular way. And so what that means is that these cells will become better at resisting deformation in a particular direction. I hope that makes some sense. So I've given you a couple glimpses. There's more complexity behind all of these, but we only have so much time, in how the cell wall mechanics might be involved in sort of how we can measure it and how it might be involved in cell behaviors, but also some hints as to how you might sense force if something was happening around you in terms of changing forces due to other cells growing or your own shape. I think I can do this really quickly. So here's my short example of how all of this could come together to help you understand cell expansion. So Charlie mentioned how materials get to the cell wall, but also how proteins that could change the cell wall would get there. And I'm going to bring in, they come from the Golgi, right? But that actually has to do also with the cytoskeleton. So Golgi trafficking happens through the actin cytoskeleton. I hope that Ram was introducing this to you yesterday. And as I just mentioned, cellulose synthase, once the complexes are in the membrane, they actually track along microtubules, and that'll change the orientation of fibers, which can change mechanics. So my short example here is about how we take a sort of relatively sort of square cell and get it to elongate more in one direction than another. And this is really important for young seedling emergence from the soil, but actually in a lot of other contexts in plant growth as well. So one of the things that we found in the lab is that if you change, and it was kind of weird for us, but if you change the gelling capacity of the pectin gel, we could actually change the elongation of these hypocotyls. And so what we've done here is we've induced an enzyme that allows the pectin to cross-link more, or an enzyme inhibitor that stops the pectin from being able to sort of cross-link well here. And if we allow for more cross-linking to occur, the organ is shorter. If we allow for less cross-linking to occur, the organ ends up being longer. And we could see the same thing on cell levels. I don't think my movie is going to play for you, but essentially this one grows faster than this one just by changing the pectin matrix mechanics, by changing the gelation. So this one should give us a more rigid gel, and this should give us a more soft gel. And we were able to actually use AFM to show that that appeared to be true, at least with respect to the elasticity of the cell walls. Oh, there go my movies. But you don't get to see them for time, sorry. When we looked at cell levels, we could actually use immunohistochemistry, using antibodies for different types of pectin. We could see that there were differences in individual cells. I'm going to kind of show you really quickly here. So this antibody should mark softer pectin that's less able to calcium cross-link, less able to gel. And this is a section of a hypocotyl. And in these epidermal cells, what we could see is that there was more of that on the walls that were elongating. So growing more, you would have more soft type of pectin, versus this antibody, which shows us where the pectin can cross-link. And it was more on these walls here that sort of control the width maybe of the cells. Using AFM, again, so here's a little picture of what that might actually look like. What we could see is that we could actually see differences in mechanics between the walls that were elongating and the walls that were not, where those pectin markers were. So elongating walls had a lower elastic modulus than the walls that sort of controlled width, where we had that stiffer pectin. So there's a good correlation here. We did some fancy modeling, but I'm not going to tell you too much about it because I want Charlie to tell his story. So I'm going to stop. Okay.
Any questions for Siobhan before I jump in?
If you have some that haven't been addressed, you can always, contact us. We can Zoom later. You can send emails. Charlie and I are both super open to that. So just so you know, especially as you work on your projects, if you have more questions, okay?
All right, so I'm going to spend just a couple of minutes talking about how we're trying to understand the mechanobiology of the cell wall in stomatal guard cells. And we have two RU students working on this project as part of the CMB, Emily and Mythili. So stomata are pores in the surface of plants that can open and close to control the uptake of CO2 and the loss of water from the plant surface. And they have very unique cell wall structures in that the cellulose is wrapped radially around the guard cells that flank each stomatal pore. And we think that that forces the cells, when they're swelling, to drive stomatal opening to elongate and forces those cells apart such that they open the pore. And then when the pore closes, those cells shrink and collapse the pore back to being closed, as you can see in the lower right here. So we've done some cell biological work to understand how cellulose changes in its organization between the open and closed states, which is what you can see in this little mouth talking video here. So we've also tried to understand the function of a class of enzymes called polygalacturonases that cleave the backbone of homogalactronan, that simplest form of pectin. And this was work that was done by a great PhD student, Yua Rui. So we knocked out this polygalactronase in Arabidopsis. We can actually order knockouts through the mail if we work in Arabidopsis. And we saw that those plants were smaller than wild type. When we overexpressed this polygalactronase, which we called polygalactronase involved in expansion 3, or PGX3 for short, we saw that the plants were a little bit larger than our wild type controls over here on the left. So we performed stomatal function assays by inducing the stomata to close or inducing them to open. And the interesting thing is that we saw differences in the closure behavior in the knockout and in the opening behavior in the overexpression line. So in wild type plants, if you induce stomatal closure with a hormone called ABA, the stomata close smoothly over time. But in the knockout, you can see this sort of stepwise pattern of closure. And in the overexpression line, we saw accelerated stomatal opening in response to a drug that drives protons out of the cell and causes the stomata to open. So we also did some time-lapse imaging of stomata in wild type and PGX3 knockout plants. And you can see on the left that when we induced stomatal closure with, again, this hormone ABA, we saw that most of the cells closed over time. But some of the PGX3 knockout stomata performed aberrantly. They would actually close and then open, or open and then close. So they had abnormal behavior, and they closed to a lesser degree than we saw in wild-type controls. So what we were able to do with this and many other experiments that I'm not going to talk about due to time is to develop a working model for how this polygalactonase might be tuning the pectin in the cell wall of these guard cells to allow them to open and close normally. So in wild-type plants, we think that the polygalactonase is tuning the size of the pectin molecules in the cell wall. So pectin is shown in blue here, cellulose is in red. And it tunes the size such that there's a balance between pectin cross-linking by calcium and pectin degradation by these polygalactonases, such that they're able to open and close smoothly. In the knockout, we think that there's less PG activity, less polygalactonase activity, more pectin cross-linking potentially, and thus a propensity for closure to behave in this stepwise fashion. And in the overexpression, we think that there's less pectin because there's more enzyme that degrades pectin, and the wall might be a little bit more fluid and allow for opening to happen faster. So what these experiments revealed to us was something that we call cryptic hysteresis, which basically is a fancy way of saying that the guard cells act in an asymmetric fashion depending on the pectin modification that's happening in their cell wall. And we're exploring that further using some nanoindentation analyses in collaboration with Joe Turner at the University of Nebraska. So we've also generated what are called finite element models of stomata in wild type and pectin deficient and other conditions where the cell wall is altered. And we found that the shear modulus in particular in these pectin deficient guard cells might be responsible for their propensity to open wider in response to stimuli that induce opening, like light or the drug fusicoccin that I introduced earlier. So it's really this shear modulus that is happening between layers of the cell wall or between directions of elastic modulus in the cell wall that we think is altered by changing the pectin in this case, and that drives stomatal opening to a larger degree. I would also point out that other groups are engineering stomata to enhance plant performance. So here they actually used an engineered channel, potassium channel, and potassium channels are one of the things that drives stomatal opening. They engineered that to be light responsive and found that the plants were able to use water more efficiently when they put in this engineered potassium channel. So it's a pretty cool way of modifying the efficiency of the plant. Okay, so I'm going to stop there because it's 12 o'clock right now. And if there are any questions from the audience, you can feel free to chime in, or again, like Siobhan said, I'm more than happy to answer questions post-presentation. It's very quiet out there in Zoom land.
You probably want to have a chance to have a break before Guy's going to start the next lecture. So
thanks both of you. Actually very nicely connected with things that I had talked about with respect to the cytoskeleton, as well as I think what Anders had talked about, about forces and things. So yeah.
Cool. Awesome. Well, thanks everybody for being here. Thanks also to the RU students for joining, and send us emails at any point when you start having questions. We're happy to answer anything we can.